Methotrexate is a chemotherapy drug used in the treatment of cancer. It closely resembles folic acid, a coenzyme used in the synthesis for nucleic acids and amino acids. Methotrexate is shown here with its carbon atoms colored in green. It can bind to proteins that otherwise would accommodate folic acid. Here you see methotrexate binding snugly into the pocket of dihydrofolate reductase. Dihydrofolate reductase turns dihydrofolate into tetrahydrofolate. In order to do so, it needs another coenzyme known as NADPH. NADPH transfers a hydride, shown here in red, onto dihydrofolate. The enzyme dihydrofolate reductase hence also needs to accommodate NADPH. The carbon atoms of NADPH are shown here in yellow to distinguish it from methotrexate, which is also bound and shown in green. The enzyme dihydrofolate reductase closely and exactly positions NADPH and methotrexate to allow for the hydride transfer. Let's have a closer look at the interactions that are necessary to position the two molecules so exactly. There are electrostatic interactions, like the one you see here with an arginine interacting with a carboxylic acid group from methotrexate. Also hydrogen bonds between a carbonyl group and an amine group of a side chain, and several hydrophobic interactions as well as aromatic stacking interactions play a role in the positioning of the two molecules. Lastly, here you see the phosphate group of NADPH with numerous hydrogen bonds to protein components. In this structure, NADPH and methotrexate snugly fit into the binding site. We can depict this binding by choosing CPK models and rendering the complete protein with CPK atoms. Or what you often also see are ribbon diagrams of protein structures and molecules of interest highlighted as CPK or ball and stick models. To have a closer look at the architecture of proteins, ribbon diagrams can be very helpful. Here the secondary structure elements, alpha helices, are colored in red, and the beta strands forming a long beta sheet are color-coded in green. The analysis of protein structures, such as dihydrofolate reductase, and the exact mechanism and interactions of incorporation of drug molecules such as methotrexate can make huge contributions to the design of new and improved drugs for the treatment of various diseases, including cancer.